Dear viewers, this video is part 5 of the 7-part series on lymphocytes and immunity in the Medicine for Anyone in the YouTube channel. Mechanism of action of antibodies. Antibodies act mainly in two ways to protect the body against invading agents. One, by direct attack on the invader, and two, by activation of the complement system that then has multiple means of its own for destroying the invader. This picture shows binding of antigen molecules to one another by bivalent antibodies. Direct action of antibodies on invading agents. Figure binding of antigen molecules to one another by bivalent antibodies shown earlier. Please refer to it. Binding of antigen molecules to one another by bivalent antibodies shows antibodies designated by green Y-shaped bars reacting with antigens designated by the red-shaped objects. Because of the bivalent nature of the antibodies and the multiple antigen sites on most invading agents, the antibodies can inactivate the invading agent in one of several ways as follows. 1. Agglutination, in which multiple large particles with antigens on their surfaces, example bacteria or red cells, are bound together into a clump. 2. Precipitation, in which the molecular complex of soluble antigen, example tetanus toxin, and antibody becomes so large that it is rendered insoluble and precipitates. 3. Neutralization, in which the antibodies cover the toxic sites of the antigenic agent. 4. Lysis, in which some potent antibodies are occasionally capable of directly attacking membranes of cellular agents and thereby cause rupture of the agent. These direct actions of antibodies often are not strong enough to play a major role in protecting the body against the invader. So, dear listeners, by the way, this educational video is made possible by PrescribeOn, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribeon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Complement is a collection of about 20 proteins which are enzyme precursors. They are designated from C1 through C9, B and D. They are present normally among plasma proteins in blood. Most of the protection occurs through the amplifying effects of the complement system as follows. The figure complement pathways and effector mechanisms shown earlier, kindly refer to it. The main function of the complement system is to enhance, complement the actions of antibodies and phagocytic cells in neutralizing and destroying pathogens, removing damaged cells from the body, and promoting inflammation. See the figure again. Complement is a collective term that describes a system of about 20 proteins, many of which are enzyme precursors. The principal actors in this system are 11 proteins designated C1 through C9, B and D. All these are present normally among the plasma proteins in the blood, as well as among the proteins that leak out of the capillaries into the tissue spaces. The enzyme precursors are normally inactive, but can be activated by the so-called classical pathway. The classical pathway is initiated by an antigen-antibody reaction. That is, when an antibody binds with an antigen, a specific reactive site on the constant portion of the antibody becomes uncovered or activated, and this in turn binds directly with the C1 molecule of the complement system. This sets into motion a cascade of sequential reactions, beginning with activation of the proenzyme C1. The C1 enzymes that are formed then activate successively increasing quantities of enzymes in the later stages of the system so that, from a small beginning, an extremely large amplified reaction occurs. Multiple end products are formed, and several of these have important effects that help prevent damage to the body's tissue caused by the invading organism or toxin. Among the more important effects are the following. 1. Opsonization and phagocytosis. One of the products of the complement cascade, C3b, strongly activates phagocytosis by neutrophils and macrophages, causing these cells to engulf the bacteria to which the antigen antibody complexes are attached. This process is called opsonization. It often enhances the number of bacteria that can be destroyed by many hundredfold. 2. Lysis. One of the most important of all the products of the complement cascade is the membrane attack complex, also called the cytolytic complex, which is a combination of multiple complement factors designated as C5b6789. This membrane attack complex inserts itself into the lipid bilayer of the cell membrane, creating pores that are permeable to ions and causing osmotic rupture of the cell membranes of bacteria or other invading organisms. 3. Agglutination. The complement products also change the surfaces of the invading organisms, causing them to adhere to one another, thus promoting agglutination. 4. Neutralization of viruses. The complement enzymes and other complement products can attack the structures of some viruses and thereby render them non-virulent. 5. Chemotaxis. Fragment C5a initiates chemotaxis of neutrophils and macrophages, thus causing large numbers of these phagocytes to migrate into the tissue area adjacent to the antigenic agent. 6. Activation of mast cell and basophils. Fragments of C3a, C4a, and C5a activate mast cells and basophils, causing them to release histamine, heparin, and several other substances into the local fluids. These substances in turn cause increased local blood flow, increased leakage of fluid and plasma protein into the tissue, and other local tissue reactions that help inactivate or immobilize the antigenic agent. These same factors play a major role in inflammation, discussed in WBCs, granulocytes, and in WBCs granulocytes functional aspects in Medicine for Anyone YouTube channel. 7. Inflammatory effects. In addition to inflammatory effects caused by activation of the mast cells and basophils, several other complement products contribute to local inflammation. These products cause the already increased blood flow to increase still further, the capillary leakage of proteins to be increased, and the interstitial fluid proteins to coagulate in the tissue spaces, thus preventing movement of the invading organisms through the tissues. Special attributes of T lymphocyte system, activated T cells, and cell-mediated immunity. Release of activated T cells from lymphoid tissue and formation of memory cells. On exposure to the proper antigen, as presented by adjacent macrophages, the T lymphocytes of a specific lymphocyte clone proliferate and release large numbers of activated specifically reacting T cells in ways that parallel antibody release by activated B cells. The principal differences is that instead of releasing antibodies, whole activated T cells are formed and released into the lymph. These T cells then pass into the circulation, are distributed throughout the body, pass through the capillaries walls into the tissue spaces, back into the lymph and blood again, and circulate again and again throughout the body, sometimes lasting for months or even years. Also, T lymphocyte memory cells are formed in the same way that B memory cells are formed in the antibody system. That is, when a clone of T lymphocytes is activated by an antigen, many of the newly formed lymphocytes are preserved in the lymphoid tissue to become additional T lymphocytes of that specific clone. In fact, these memory cells even spread throughout the lymphoid tissue of the entire body. Therefore, on subsequent exposure to the same antigen anywhere in the body, release of activated T cells occurs far more rapidly and much more powerfully than during the first exposure. This picture shows activation of T cells requiring interaction of T cell receptors with a foreign protein that is antigen, that is transported to the surface of the antigen presenting cell by a major histocompatibility complex MHC protein. Cell to cell adhesion proteins enable the T cell to bind to the antigen presenting cell long enough to become activated. Antigen presenting cells, major histocompatibility complex proteins, and antigen receptors on T lymphocytes. T cell responses are extremely antigen specific, like the antibody responses of B cells, and are at least as important as antibodies in defending against infection. In fact, acquired immune responses usually require assistance from T cells to begin the process, and T cells play a major role in helping eliminate invading pathogens. Although B lymphocytes recognize intact antigens, T lymphocytes respond to antigens only when they are bound to specific molecules called MSC proteins on the surface of antigen presenting cells in the lymphoid tissues. See the figure T cell antigen presenting cell interaction shown earlier. The three major types of antigen presenting cells are macrophages, B lymphocytes, and dendritic cells. The dendritic cells, also known as accessory cells, the most potent of the antigen presenting cells, are located throughout the body, and their main function is to present antigens to cells. The interaction of cell adhesion proteins is critical in permitting the T cells to bind to antigen presenting cells long enough to become activated. The MHC proteins are encoded by a large group of genes called the major histocompatibility complex, MHC. The MHC proteins bind peptide fragments of antigen proteins that are degraded inside antigen presenting cells and then transport them to the cell surface. There are two types of MHC proteins. One MHC1 protein, that is presenting antigens to cytotoxic T cells, and two MHC2 proteins which present antigens to T helper cells. The specific functions of cytotoxic and T helper cells are discussed later. The antigens on the surface of antigen presenting cells bind with receptor molecules on the surfaces of T cells in the same way that they bind with plasma protein antibodies. These receptor molecules are composed of a variable unit similar to the variable portion of the humoral antibody, but its system's stem section is firmly bound to the cell membrane of the T lymphocyte. There are as many as 100,000 receptor sites on a single T cell. Different types of T cells and their functions. It has become clear that there are multiple types of T cells classified into three major groups. 1. T helper cells, 2. Cytotoxic T cells, and 3. Regulatory T cells, also called suppressor T cells. The functions of each of these T cells are distinct. T helper cells are the most numerous T cells. This picture shows the regulation of the immune system. On presenting with a specific antigen, the CD4 positive T helper cells produces different types of lymphokines stimulating CD8 positive cytotoxic T cells, the so-called killer cells, stimulates suppressor T cells, which are regulatory cells, which are CD4 positive, and also stimulate B cells, which become plasma cells to secrete appropriate immunoglobulins. Figure immune system regulation. Kindly see the picture shown just earlier. The T helper cells are the most numerous of the T cells, usually constituting more than 75% of all of them. As their name implies, they help in the functions of the immune system in many ways. They serve as the major regulator of virtually all immune functions, as shown in the figure shown earlier. They do this by forming a series of protein mediators called lymphokines that act on the other cells of the immune system as well as on bone marrow cells. When stimulated, naive CD4 positive T helper cells can differentiate into subsets that produce different lymphokines and perform different functions. Specific regulatory functions of lymphokines. In the absence of the lymphokines from the T helper cells, the remainder of the immune system is almost paralyzed. In fact, it is the T helper cells that are inactivated or destroyed by the human immunodeficiency virus HIV, which leaves the body almost totally unprotected against infectious diseases, therefore leading to the now well-known debilitating and lethal effects of acquired immunodeficiency syndrome, that is so-called AIDS. Some of the specific regulatory functions are described in the following sections. Stimulation of growth and proliferation of cytotoxic T cells and regulatory T cells. In the absence of T helper cells, the clones for producing cytotoxic T cells and regulatory T cells are activated only slightly by most antigens. The lymphokine interleukin 2 has an especially strong stimulatory effect in causing growth and proliferation of cytotoxic and regulatory T cells. In addition, several of the other lymphokines have less potent effects. Stimulation of B cell growth and differentiation to form plasma cells and antibodies. The direct actions of antigens to cause B cell growth, proliferation, formation of plasma cells, and secretion of antibodies are also slight without the help of the T helper cells. Almost all the interleukins participate in the B cell response, but especially interleukins 4, 5, and 6. These three interleukins have such potent effects on the B cells that they have been called B cell stimulating factors or B cell growth factors. So, dear viewers, this is the end of part 5 of the 7-part series on lymphocytes and immunity in the Medicine for Anyone in the YouTube channel. Thank you for watching the video.